that'll help me at all. I'm still having trouble. So, I don't know if you can see some of this. Some of the particles are breaking out on the right side, and then they're dissolving. And over here on this side, they are not. You're getting a lot of powder there in the center when I stir it. So I know I have some large pieces on the right. But I don't have the, and you see the solution is sort of clear. Here on the left, I got, and when I did manage to break it up, all that stuff is not dissolving. I got a whole lot of sludge and a little cloudier sample over there. This is just light. That's not precipitate. It might be a little better. So it does dissolve better at the high pH than at the low pH. And a problem then is people taking an overdose of aspirin have all this crud sitting in their stomachs because it's the protonated form, the COOH, which is less soluble than an ion. Now this is a little bit of the, you know you have stomach acid in your stomach. This is anywhere, that's 0.01 molar is pH 2. And in the intestine, you have a neutral pH. So what's the kind of acid that the stomach secretes that makes stomach acid that makes it low pH? It's hydrochloric acid. So when that solution then goes into your intestine secretes a base, which neutralizes the stomach acid. Do you know what kind of base your intestine secretes? It's not sodium hydroxide. What? Yeah, it's bicarbonate, sodium bicarbonate. And you remember the bicarbonate can react, they can absorb an H plus and make the H2CO3, which can make water and carbon dioxide. So that's your biological base. Now what people did, if people have an overdose of a kind of aspirin that's just going to sit in your stomach with all that sludge, like we were demonstrating here in class, sort of, they could be taken to an emergency room and have a tube put through their nose down into their stomach and then wash out their stomach. And what they would use is bicarbonate. It's the same kind of base that your own body makes to help neutralize stomach acid. And this would then raise the pH and the aspirin would become more soluble when it was in the deprotonated form. Remember the pK was 3. So if you could get your pH to 7 with this base, then the aspirin would dissolve better. It's called a lavage, a stomach lavage. And the root word for this is a Latin word which means to wash. So you're washing your stomach. And you know the word lavatory, it's both where we excrete waste and where we bathe ourselves. So this is a word for bathing. Now, I went to the pharmacy yesterday to try to find aspirin with no coating on it so I could get it to dissolve a little better or faster. And I found out that every single brand at the pharmacy has a coating on it. And some of them are talking about an enteric coating. Now the idea here is, especially for people that are on an aspirin regimen, they're supposed to take aspirin, and you don't want that sludge in the stomach. This is a kind of coating that we'll look at what it says here on the internet site. What is meant by enteric coating? And it says it's a delayed release safety coating that provides added stomach protection. It's designed to let the aspirin tablet pass through the stomach to the small intestine before it dissolves. So it would take longer for the aspirin to be absorbed. If you need aspirin for a headache right away, this would not be so good. But the people who need to take aspirin on a regular basis, there will be less irritation to the stomach. That coating should really resist dissolving. Now let's look at the chemistry of it. These are some of the compounds that are used in that coating. You either use this, the upper compound is from seaweed. Now that's a carboxyl group, all right? That's the low pH form, the form it would have in, both of these are drawn as carboxyl groups, the form you would have in the stomach. And this chemical, which looks like it is a sugar polymer, but it has a carboxyl group on it. The CH2OH sticking out has become a COOH carboxyl group. The lower one is just a fatty acid. And these coatings in the stomach will be carboxyl insoluble, but in the intestine become carboxyl minus. And these large molecules are especially resistant to break down, and so they'll keep the capsid in that tablet or capsid form, the pill form, and protect the ingredients inside, the aspirin inside, from being exposed and making that yucky looking sludge. And then you won't have this problem of a pool of stuff sitting down there in your stomach somewhere. All right, so that's a little demonstration here of pills where we have a combination of the aspirin. I should maybe buy a bottle of pure acetylsalicylic acid from an organic chemistry shop. And then and maybe have that for my letter variation. All right, now what are some other stories that are particularly interesting about solubilities? And I pick and choose my next favorite stories. They're about protein sequences and some patterns that are observed. All right, so let's go to figure 15 as the first of these in this group. Maybe that was back a ways. This is a protein that is secreted, it's made in our pancreas. Our pancreas is where we make trypsin, one of these digestive enzymes we talked about. And it's actually, in the pancreas, it's made as trypsinogen, a precursor of the enzyme. That precursor is transported from the pancreas to the intestine, and it becomes activated in the intestine, and then just digests food there. Now, if that enzyme became activated in our pancreas, they might start eating up the walls of the pancreas, which are not protected from enzymes the way the intestinal walls are protected. So, as a safety device, our pancreas also secretes this little molecule which binds to trypsin if anywhere made in the pancreas, anywhere activated, and prevents trypsin from working. It's a small protein. Now, I want to focus your attention to this helix, which is sort of on the back side of this molecule, but at the very far side, the very far back, is facing water. And the other side is more towards our eyes, is facing that white helix above it. And it's buried. So as we travel around and around and around and around the sequence, we'll have some amino acids that are in the inside and then some on the outside, and then some on the inside, and then some on the outside. And if you think about the kind of sequence that would be likely to do it, it would be something that would have some non-polars to be on the inside, and then some polars on the outside, and we're going around the helix again, some non-polars on the inside, and comes around again and have polars on the outside. So we're going to look at a plot of those amino acids. I know you don't know the 20 kinds yet, so I sort of drew a little bit of their structures off to the side. And I colored in white the types that are usually exposed to water in our various proteins, and almost always exposed are the ions. And here you'll see them. And you'll notice that there's a patch of negative ones and a patch of positive ones. They're 3 to 4 residues apart. I then colored in black those whose atoms are hydrogen, carbon, and sulfur, which we commented on Wednesday, have the same electronegativity value. So that 2.1, 2.3, so that the bonds between them are nonpolar. So I colored those in black. And you'll see those are also spaced about that same distance apart. Now the story here is that, here's, our next slide sort of says how many amino acids does it take to go around one time to make the trip around. This is a plot looking down the axis of alpha helix in a protein. So if this is residue 1, the next one is 100 degrees around, then 200 degrees around, then 300 degrees around. And if we did one more, it would be over here at 400 degrees. And so it takes between 3 and 4 residues to go around one time. If you think what that's going to, and that's exactly this spacing here, between 3 to 4 residues to go around the wheel one time. So now what I did was take that sequence, and instead of writing the names of the amino acids, I just sort of copied in these symbols going around and around and around. So we start out with a striped one, sort of in between. We get to these 2 negative ions, the white ones on that side. Then we go around 51 and 52 are black-colored, nonpolar. We come around again, 53 is a positive charge, then a polar one, a striped ball. Then we get to black on the other side. And what you see is that one side of the helix is nonpolar, and the other side of the helix is polar, including ions especially. And that's exactly the kind of a sequence that would give this feature of having a helix with one surface, not one end of the helix, but one surface of the helix facing water and one surface being buried. It's part of the strategy of designing a sequence that will give a particular three-dimensional conformation in the folded protein. So that's an interesting polar-nonpolar story, I think. That pattern and its consequences. Now let's talk about one particular kind of virus. Not all viruses are this simple, but this particular one has a really simple pattern to it. So what I did was color the positive side. This is a small virus, 50 amino acids here. And I colored in black the amino acids whose side chain are negative. And I colored in white the amino acids whose side chain are positive. And the sequence is sort of cut into thirds. The first third is mostly negative charges. The middle third is mostly nonpolar. And the last third is mostly positive. Now, things aren't always that way. I mean, here's a positive, 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 positive. All right, and these are negative, 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 etc. That's just a simple pattern. Well, what's the consequence of that, and how might that help? The DNA gene of the virus is packed inside the virus particle. And DNA has negative phosphate groups on it. So what is complementary to that, and could sort of wrap itself around the DNA or interact favorably with it, would be these positive ends of the protein. So you see that third interacting well with the negative DNA. Then you got some nonpolar parts in the middle. Well, those are the parts that will get buried as you form this layer. And the other end of the protein, which would tend to be repelled by the DNA, will interact well with water because it's an ionic, but it's going to be repelled away from the DNA. And that end then faces the outside. So it's an example of one virus where the pattern is particularly simple and we can rationalize why that arrangement might work. Now let's talk about a human disease called sickle cell anemia. Our red blood cells, the normal shape of them is a little disc. This is a picture of red blood cells. The figures that are going to follow are right in order 18. I have put this at UB Learns, or I will have. So this is a red blood cell, an erythrocyte. And they're small or circular, and they can go through our tiny capillaries that are just a little bit wider than one red blood cell. They can squeeze through that. This is the red blood cell of someone with sickle cell anemia. And you see these long fibers sticking that have formed in this cell. Some of them have actually pushed out the side here in this photograph. These are fibers of hemoglobin. Hemoglobin is not supposed to make fibers. But in this disease, each hemoglobin molecule binds to another one, to another one, to another one, makes these long fibers which stretch out and make the cell have a shape called sickle shape. I wonder if any of you ever had one of these. We used to have at a cabin we owned in the woods in New Hampshire, these sickles. There'd be a long wooden handle and then a cutting blade. And you could go and slash, wave that back and forth and cut down big weeds or grass that had grown up in the sand. And that's a tool called, a gardening tool or agricultural tool, a sickle. So some people think this long handle and this object at the end is sickle shaped, so it's called sickle cell anemia. Anemia because we're eventually going to lose our hemoglobin and not be able to transport oxygen and have trouble breathing. Now, the mutation that occurs is the normal type has a polar side chain on its surface, but in the mutant, it becomes a valine, which is all hydrocarbon. So glutamic acid has 2 CH2s on it, but ends in carboxyl minus. And there's a mutation in the gene that codes for that creates a different codon, a different triplet in the messenger RNA, which codes for valine. Now, our hemoglobin molecules also sort of have a nonpolar patch. And that's only a problem then as if the hemoglobin molecule also has a nonpolar protrusion or bump in it. In the wild type protein, this would be a glutamate and it would not stick into that little depression because you're not going to bury a carboxyl group. But in the mutant, then that little protrusion, which then is valine and is nonpolar, will stick into the depression of another molecule next door, and then they start forming these, this is attached to this, which bonds to this, which bonds to this, and make these long fibers that distort the red blood cells. They cannot squeeze through our capillaries, so they get broken. The water pressure is trying to push them into the capillaries. It breaks open those sickled cells, the hemoglobin is released, other parts of the body reabsorb it, they think it's not correct, and then you don't have enough red blood cells in your blood. And this can also be very painful, this blockage of capillaries, and so that's a human disease. We'll talk more about it maybe in part 2 of the course. Maybe not. We'll talk a little bit more about it. I'm not sure if I'll do it. I do have a medical supplement. I'm not sure if I'm going to use that one or something else later in the course. All right. Now, we have a few minutes left. Let's talk about a biological detergent. I'll tell you in a minute. Where we're going here is figure 23. Oh no, that's not it. What's figure 24? Again, maybe that's not it. What about salt? All right, this is a picture of bile 26. Now your gallbladder secretes bile. The bile is made in the liver. It's moved to your gallbladder, which stores it. And then when you eat a meal that has a lot of fat in it, which is, pretty insoluble stuff, your body senses that and sends a signal to the gallbladder, release these chemicals, these bile salts they're called, to your intestine to help solubilize the lipids. Now a detergent is any molecule that has sort of a polar and a non-polar end. Your soap, your laundry detergent, what you bathe yourself in, you lavage yourself in the laboratory, they have a polar and a non-polar end to it. Now most of this is non-polar, except for out here, where in some cases, let's see, we have an R1 group and an R2 group. The R1 group is usually like this. Here we have some R2 groups. You see an acid group, sulfonic acid, you can get SO3 minus, COO minus. There are some polar portions. Now actually these polar portions tend to lie on one side. I'm going to show you I think I might have a three-dimensional drawing. I post this at UB Learns also. You see the OHs all pointing towards the back in these steric drawings, these little dotted wedges going to the OHs. And furthermore, this carboxyl group has a long arm on it, and it can stick on any side at once then. And it reaches back to the same side where the OHs are. And so we have this sort of planar four-ring structure with hydroxyl groups and negative carboxyl group on one side, and the other side is more nonpolar. So what happens then is your detergent mixes with your fats, and the nonpolar side of the detergent will sort of coat or aggregate with the nonpolar fats, and the polar side of the salts, those COO minus or SO3 minus and hydroxyl groups, that'll face out towards the water, which it likes. So the polar side faces the water, the nonpolar side faces your lipids, and it then makes little droplets like this where your fat can be broken up into smaller units that then can be processed by enzymes on the surfaces of your intestine. So your bile is a biological detergent. All of these are short stories about polar and nonpolar groups, thinking about pH sometimes. And there's one or two more: poison ivy toxin, a bug that walks on water, several more in your lecture, which you'll be able to read. They're sort of simple to understand. So I hope you like this lecture. We're going to do kinetics and equilibrium.